Hello, this is Mr. McLean. In today's discussion, we're going to be heading into the idea that natural selection is going to act on phenotypes. And probably one of the biggest ideas we're going to be heading into is the concept behind Hardy-Weinberg equilibrium. So I'll take you through that today. All right, so getting us started. All right, so I guess probably one of the first points that we should make sure that we're all on the same page with is understanding that what the concept of a population is. And when we look at a population, we're talking about organisms that happen to be of the same species living in a particular area at a certain set time. Okay, so these are all members of the same species. And one thing that's going to be really critical to take a look at is that populations will evolve, individuals will not. We'll definitely be emphasizing that. So part of today's objective is to introduce some of the tools that can be used to evaluate a population to show whether or not it actually is evolving. Um, so one of the videos that I have embedded in my lecture deals with the rock pocket mouse, and I really suggest that you look at this. We'll have an assignment that goes along with it. It's linked to a YouTube video in there, but it's an excellent example of a population that definitely underwent some change based on phenotypes that were favorable for the new environment that happened after an environmental change, you could say. All right, so that we're all on the same page, some definitions here. We got a population. We'd say it's a localized group of individuals that are capable of interbreeding and producing fertile offspring. So basically, that's the biological definition of a species. So biologically, they have to be able to have fertile offspring between the two. As we move down, we have the term gene pool, which would indicate all of the possible alleles for a particular gene within that particular population. So, if we were taking a look at something like in the example of the video with the rock pocket mouse, it was a gene that affected the fur color on the mouse. So if we go through there and we take a look at the entire population, what alleles do each of the individuals contribute to that? And the grand total of all those alleles makes up our gene pool. So gene pools definitely can change as the population changes. And then when we talk about a locus, we're talking about a particular location within the genome. Okay. And it says a locus is fixed if all the individuals in the population are homozygous for the same allele. So if we have a situation where there's no diversity of the alleles, so in many of our genetic studies, we were looking at things that were going to be following simple Mendelian inheritance patterns. So if everything, let's say, was the dominant allele and there were no recessive alleles in there, we would say that that's a fixed locus because everybody's exactly the same. Now, should there be a mutation or some introduction from an outside population, then that would change. All right. Okay. So when we talk about the frequency of an allele, basically what that's saying is what's the percentage that that allele appears in our population. So the math behind it's really simple. So when we look at organisms that are diploid, so organisms that are sexually reproducing, the total number of alleles for a locus is the total number of individuals times two, because we all contribute two alleles for every locus, because we have two chromosomes for each set, so we're diploid. So the total number of dominant alleles at a locus is two alleles for each homozygous dominant, plus one allele from each heterozygous individual. And then the same applies for the recessive. So basically, all of your individuals that are homozygous dominant, so if I had a population and I had 100 individuals that were homozygous dominant, I would actually be contributing 200 alleles. And then if I had, let's say, 50 people that were heterozygous, I would get 50 more dominant alleles because half of their alleles are going to be dominant and half of them are going to be recessive. And then, we can apply that same logic for the recessive. So if I said I had 100 recessive, homozygous recessive, you would actually have 200 recessive alleles plus the 50 that were heterozygous. Okay, so this is going to get us into the Hardy-Weinberg component of it. So this is where we introduce some of the variables, and we just want to make sure everybody's on the same page with this. Um, so it says if there are two alleles at a locus, we choose to use the variable P and Q to represent the frequencies. So P represents the percentage or frequency of dominant alleles. Q represents the frequency or percentage of recessive alleles. So it says the frequency of all the alleles in a population has to add up to 1, which would mean that the percentage of all the dominant and the percentage of all the recessive would equal all of them, which makes sense. So we see this underlying equation right here, and this is going to be part of our understanding of how alleles work in a population. So all of the dominant alleles in terms of the percentage. So if I said, hey, I got 40% of dominant and I've got 60% of recessive, I have 100% of all my alleles. So the P and the Q represent the allele frequency, or the percentage of the time that that allele shows up. So how that ties in with one of our most important calculations in AP Bio is the idea of the Hardy-Weinberg principle. And basically, it describes a population that's not evolving. Okay, so the Hardy-Weinberg concept is a mathematical concept that you can evaluate a population, and it essentially will tell you what a non-evolving population will be. And we're going to go through those criteria. And if it fails to meet the criteria, we can therefore conclude that the population is evolving. All right, so basically, it's a way for us to look at a population and mathematically have some evidence that yes, there's evolution, or no, this population is not changing. So let's take a look at this. The Hardy-Weinberg principle states that the frequencies of alleles and genotypes in a population will remain constant from generation to generation. All right, so a couple things. It does not say the phenotypes will stay constant. We're going to see how these can kind of get mixed up here, but it says the frequency of the alleles are going to remain constant. In a given population where gametes contribute to the next generation randomly, allele frequencies are not going to change. So if you had 60 and 40% in the initial generation, and if there's no evolution going on, it's going to remain 40 and 60 every generation after that. So the idea is they're going to get shuffled. So when we see this last statement that the Mendelian inheritance preserves genetic variation in a population, those alleles might still be in the same ratio, but the way that they combine to form the actual phenotypes is going to potentially be different. All right, so let's take a look at this. They use the example of this collection of, we'll call them gumballs in this gene pool right here. So the red is going to represent the red allele, which in this particular case we're going to say is the dominant allele for the sake of this particular problem. And the Q is going to represent our white, which is going to be the allele for the recessive phenotype, or the recessive color in this case here. So this represents our gene pool. This is the pool of all of the different alleles that are in there. So when we go through here and we take a look at it, it's telling us that out of this gene pool, 80% of these are red, 20% of these are white. So when we go to form the gamete, whether it's the egg or the sperm, those ratios are going to hold true. It's going to be an 80-20 split on those things. Okay? So let's talk about how that relates to the Hardy-Weinberg equation. So Hardy-Weinberg equilibrium, which was determined, by the way, by a mathematician and a physician who got together and evaluated lots and lots of statistical data. And the Hardy-Weinberg equilibrium describes the constant frequency of alleles in such a gene pool. So the P and the Q represent the frequencies of the only two alleles in the population at a locus. So here's the equation. P squared plus 2PQ plus Q squared equals 1. So what this is going to do is this represents genotypes. Okay, this is a lot of times where people get kind of mixed up. You might recall we said P and Q were the actual allele frequencies, but the allele frequencies are not genotypes. So let's kind of run through this. P squared is the percentage of individuals that are homozygous dominant. 2PQ are heterozygous individuals. The percentage, whatever this value is, that's the percentage of the population that's heterozygous. Q squared, these are homozygous recessive individuals. So if you add up all the homozygous recessive, all the heterozygous, and all the homozygous recessive, so homozygous dominant, heterozygous, homozygous recessive, you put them all together, that equals everybody. And that would make sense based on the layout of these different things. Okay, so this Punnett square that's in her book, it's kind of skewed to show that there's definitely a different possibility of getting some of these different combinations. So if we take a look at it, 80% of the alleles were red in both the male and the female. So if you do a Punnett square and you weight that Punnett square, so basically we're saying this is 80% and 80%, that's going to tell me that my P squared, or my homozygous dominant, is actually going to be 64% of the population. That's why it's much larger in this picture here. If we go down here, we're going to find that we have red 80%, white at 20%, so that's going to be our 16%. And then over here, we got 80 and 20, that's 16. So this is PQ, and this is PQ. So when we want to figure out what's the total percentage of heterozygous, it's both of these. That's why we say 2PQ. So it would be 16 and 16, or 32%. Okay, and then our last grouping in here is going to be the white ones, and we have a 20% and a 20%. So that's going to give us our 4%. So when you add these up and you kind of go through here, you're going to find that this is 100% of everybody, or of all the flowers. So that's kind of how that's going to work out as we move through there. All right. So as we kind of wrap this up, and then I'll have a second video for actually solving some problems. The Hardy-Weinberg theorem describes a hypothetical population. So in real populations, allele and gene frequencies do change over time, but here's the requirements for equilibrium. 5 things. Number 1, you can't have any mutations. All right, so we know that we can't eliminate mutations, but if we have a mutation rate that's really low, we can apply this. And we'll look at some examples in the next video. You have to have random mating. So by random mating, what that's ultimately saying is that you're not clustering individuals of similar genetics together. So you want a more random opportunity for individuals to interact. No natural selection. So by natural selection, in this case, we're talking about something that's going to wipe out individuals from the population. So you don't want any of the individuals to die off because of natural selection. You want to kind of keep them in the population. It has to be a really big population. If you read some of the writing, they say infinitely large, which is really big. So the bigger the population, the closer to accuracy we'll get. And then number 5 is you can't have any gene flow, which means that individuals are not going to be able to enter into a population or leave the population. So what we'll do is we're going to stop here. And in my next video, I'll highlight these points again and we'll solve some problems. So hopefully that helps. Like I said, we're building on a concept that natural selection acts on phenotypes. Have a good day.